Somewhere in the western Pacific Ocean, east of the Philippines and south of Japan, the seafloor drops away. It drops farther than any other place on the surface of this planet. The opening is a crescent-shaped wound in the Earth's crust, stretching roughly 2,550 kilometers across the ocean floor. It is called the Mariana Trench, and tonight we go down. There is no hurry. The trench has been here for 52 million years. It was here before our species existed. It was here before our ancestors came down from the trees. It was here when the first whale song echoed through the deep Pacific and when the continents wore different faces entirely. The trench does not wait for us. It does not notice us. It simply continues. If you were to descend from the surface, the first thing you would lose is colour. Within the first 10 meters, red light vanishes. It is absorbed by the water and does not return. A little deeper and orange disappears. Then yellow. By the time you reach 200 meters, almost all visible light has been swallowed. The water around you shifts from blue to a deep indigo, and then gradually to black. By 1,000 meters, the darkness is absolute. Not the darkness of a room with the lights turned off. Not the darkness of a cloudy night. A darkness that has no boundary. No wall, no floor beneath your feet that you can see. A darkness that has never known a single photon of sunlight in the entire history of the Earth. Below that darkness, the trench descends further still. Past 6,000 meters, where the Haddle Zone begins and the pressure exceeds 600 atmospheres. Past 8,000 meters, where the last fish disappear. Past 10,000 meters, where the weight of the water above you would crush a nuclear submarine like paper. All the way down to a place known as the Challenger Deep, at the very bottom of the Mariana Trench, nearly 11 kilometers beneath the waves. It is the deepest known point on the surface of the Earth, deeper than Mount Everest is tall, deeper than most commercial aircraft fly above the ground, a place where the pressure reaches roughly 1,000 to 100 atmospheres, where the temperature hovers between 1 and 4 degrees Celsius, and where the silence is so complete that the only sound ever recorded there was the sound of our own instrument being destroyed. This is a documentary about that place, about the life that persists there, the science that struggles to measure it, and the questions it leaves unanswered. You do not need to stay awake for all of it. The trench will still be here when you return. But if you do listen, you may find that the deepest place on Earth is not the boundary of life. It may be the boundary of us. Let us begin. The first mystery of the Mariana Trench is not what lives there. It is the fact that anything lives there at all. We tend to think of extreme pressure as a mechanical problem, something that crushes, that flattens, that compresses whatever stands in its way. And at the floor of the Mariana Trench, it does exactly that. The pressure at nearly 11 kilometers of depth is roughly 100 times greater than the pressure at the surface. If you took a standard styrofoam cup and sent it to the bottom, it would return the size of a thimble, compressed so uniformly that every detail of the original cup would still be visible, just miniaturized. The polystyrene cells that once held air would have been squeezed shut. The structure collapsed into a dense, rigid pellet. That is the mechanical side of pressure, the part we can easily picture. But the harder truth unfolds at a scale no camera can capture. At those depths, water molecules are forced into spaces they do not normally occupy. Specifically, they are driven into the interior of proteins, the proteins that make up every enzyme, every receptor, every structural element in a living cell. Proteins work because of their shape. They fold into precise three-dimensional configurations, and those configurations determine what reactions they can catalyze, what signals they can transmit, what structures they can build. Under extreme hydrostatic pressure, water infiltrates those folds. The shape distorts, and when the shape distorts, the function changes. Enzymes stop behaving the way biochemistry textbooks predict. Proteins fold into configurations that no longer serve the purposes they were built for. Membranes stiffen. Reactions slow or halt altogether. This is not merely a harsh environment. It is a place where biology operates under fundamentally different physical constraints. The rules that govern molecular life at the surface do not fully apply here, and yet life persists. The answer to how it persists lies in a small molecule with a name that does not suggest anything remarkable. Trimethylamine N oxide. Scientists abbreviate it as TMAO. If you have ever smelled fish that has been sitting out for too long, you have encountered a product of TMAO's breakdown. Trimethylamine, the molecule TMAO degrades into, is the compound primarily responsible for the characteristic smell of aging seafood. It is not, on its surface, the kind of molecule you would expect to be one of the most important chemical adaptations in the history of deep-sea life. But it is. What TMAO does is simple in principle and extraordinary in effect. Under extreme pressure, it binds to the water molecules surrounding a protein. It forms strong hydrogen bonds with them, reinforcing the structure of the water itself. By stabilizing the water, it prevents that water from being forced into the protein's interior. The protein keeps its shape. The enzymes continue to function. The cell survives. In 2022, a physicist named Lorna Dugan at the University of Leeds led a study that used neutron scattering and computational modelling to examine exactly how TMAO interacts with water under pressure. Her team found that TMAO resists pressure. Induced disruption of water structure, particularly in retaining what is called the second solvation shell, the organized layer of water, molecules that surrounds dissolved substances. They calculated what they termed an osmolyte protection ratio, the concentration of TMAO needed at a given depth to effectively cancel out the destabilizing effects of pressure. And here is what is remarkable. That ratio, derived from physics and chemistry in a laboratory, matched almost exactly the concentrations of TMAO found in the muscle tissue of real organisms at corresponding ocean depths. The math and the biology converge on the same answer. The deeper a species lives, the more TMAO it carries in its body. Near the surface, most bony fish contain TMAO concentrations somewhere between 40 and 70 millimoles per kilogram. At moderate depths, a few hundred meters down, those concentrations begin to rise. By the time you reach the abyssal zone, around 2,000 to 3,000 meters, some species carry over 200 millimoles per kilogram. And at 7,000 meters, the adult snailfish carries concentrations approaching 261 millimoles per kilogram, which is nearly 4 to 6 times the concentration found in shallow water species. But even chemistry has limits. Below roughly 8,200 meters, a bony fish must accumulate so much TMAO to protect its proteins that its internal body fluids become very concentrated. The osmolality of its tissues, the total concentration of dissolved particles in its body fluids, begins to approach the osmolality of the surrounding seawater, which sits at roughly 1,100 milliosmoles per kilogram. Most shallow water, bony fish maintain their internal osmolality around 350 millismoles per kilogram, far below that of seawater. They actively pump out excess salt and retain water. But as TMAO accumulates with depth, the internal concentration climbs. At somewhere around 8,000 to 8,500 meters, the math suggests that a fish's internal osmolality would match or exceed that of seawater. At that point, the osmotic gradient reverses. Water would begin to leave the cells instead of entering them. The fish's internal balance would start to fail. The very adaptation that allowed survival at great depth would begin to work against it. This prediction was tested in 2014, when Paul Yancey and his colleagues captured 5 specimens of the Hardel snailfish Nautiloparis kermidescensis from 7,000 meters in the Kermadec Trench. These fish had the highest recorded TMAO concentrations of any species ever measured. When the researchers extrapolated the trend of TMAO accumulation with depth, they found that the concentration required for protein stabilization would push body fluid osmolality to the point of reversal at approximately 8,200 meters. That number became a kind of theoretical ceiling, the depth beyond which bony fish should not, biochemically, be able to survive, and the ocean appears to agree. No bony fish has ever been found below 8,400 meters. The deepest 25% of the ocean floor contains none at all. Only amphipods, single-celled organisms called xenophyophores, and bacteria remain below that line. The fish stop, and the silence beneath them is immense. The creature that best illustrates life at the very edge of this limit is the Mariana snailfish. Its scientific name is Pseudoliparis swairei, named after Herbert Swire, a navigating officer aboard HMS Challenger, the ship that first discovered the Mariana Trench in 1875. The snailfish was formally described in 2017 by Mackenzie Geringer and her colleagues after specimens were collected from the Mariana Trench at a depth of 7,966 meters. It has also been filmed at 8,178 meters, placing it at the very edge of the theoretical limit for fish. To look at a Mariana snailfish is to see something that barely registers as a fish at all. Its body is small, roughly the length of a human hand. Its skin is translucent, a pale pinkish white, and through that skin you can see its muscles and internal organs. It has no scales. Its skeleton is largely cartilage, soft and flexible, and its skull never fully closes. It looks, if you are being honest, like a tadpole made of tissue paper, fragile, unimpressive, easy to overlook. But within its genome lies a molecular toolkit refined by millions of years of evolution under crushing pressure. Most bony fish carry a single copy of a gene called FMO3, the gene responsible for producing the enzyme flavin-containing monooxygenase 3, which in turn synthesizes TMAO. The Mariana snailfish carries 5 copies, 5 independent versions of the gene that manufactures the one molecule standing between its proteins and destruction. It is not an accident or a redundancy. It is a precise calibration. More gene copies means more enzyme production. More enzyme production means more TMAO, and more TMAO means survival at depths where almost nothing else with a backbone can function. In September 2022, a research expedition in the Izu Ogasawara Trench near Japan filmed what appears to be a juvenile snailfish at 8,336 meters, the deepest a bony fish has ever been observed. The fish was small, estimated at 50 to 60 millimeters, and it was alone. It drifted past the baited camera and disappeared into the dark. The researchers confirmed it belonged to the genus Pseudoliparis, the same genus as the Mariana snailfish, though the exact species could not be identified from video alone. No specimen was captured. It remains for now a ghost at the edge of what vertebrate life can endure. Below that fish there is no other. Below 8,400 meters, the vertebrate world ends. What remains is a world of invertebrates and microbes, creatures whose body plans and biochemistry follow different rules entirely. Amphipods, the small crustacean scavengers that resemble aquatic woodlice, thrive at even the greatest depths. They have been found at the floor of Challenger Deep, nearly 11,000 meters down, congregating around bait in swarms of hundreds. They supplement their TMAO with another compound, psiloinositol, a molecule now being studied in Alzheimer's research for its potential to stabilize protein structures in the human brain. Between these two compounds, the amphipods manage their own version of the chemical balancing act that the fish above them can no longer sustain. And then there are the xenophyophores, single-celled organisms technically, but ones that can grow to 20 centimeters across. They build elaborate shells, called tests, from sediment particles and the remains of other organisms, cementing them together into fragile branching structures that rest on the ocean floor. They are among the largest single-celled organisms on the planet, and they have been found at the bottom of the Mariana Trench, a single cell, the size of a human fist sitting in the dark at the deepest point on Earth. The pressure paradox is not that pressure kills, it is that pressure should kill, and yet here, at the boundary where vertebrate chemistry fails, life does not end. It simply changes form. The fish give way to amphipods, the amphipods give way to microbes, and the microbes, as we will see, may have been here longer than almost anything else alive. The trench does not stop life, it reshapes it. It strips away the familiar and leaves behind only what the physics will permit. And that, quietly, is the first mystery. If pressure is the first paradox of the Mariana Trench, then evolution is the second. And it is, in some ways, the stranger one. On the surface of the Earth, the broad arc of evolution moves toward greater complexity. Over billions of years, single-celled organisms gave rise to multitellular life. Soft bodies developed hard skeletons. Simple nervous systems became brains. Eyes emerged, first as patches of light-sensitive cells, and then as the intricate optical instruments that allow a hawk to spot a mouse from a mile away. The story of life on the surface is, in its largest strokes, a story of accumulation, more parts, more specialization, more ways to sense, to move, to respond. In the Mariana Trench, the story reverses. The organisms that survive at Hadal depths have not gained complexity. They have shed it. Bone thins and softens. Pigment fades until the body is nearly transparent. Eyes shrink or vanish entirely. Organs that serve no purpose at these depths are reduced, repurposed, or deleted from the genome altogether. And this is not a sign of decay or degeneration. It is optimization. A ruthless, efficient paring down of biological systems until only what the environment demands is left. The evidence for this lies not in speculation, but in DNA. When researchers compared the complete genome of the Mariana snailfish with that of Tanaka's snailfish, a closely related species that lives in shallow water, the results were stark. 51 genes that were present and functional in the shallow species had been completely deleted in the deep sea form. Not just silenced or reduced in expression, but gone, removed from the genetic record as thoroughly as if they had never existed. Among the deleted genes was BGLAP, the gene responsible for osteocalcine, a protein essential for bone calcification. In the Hadal snailfish, this gene has been disrupted by a frame shift mutation, a shift in the reading frame of the DNA that renders the gene non-functional. The result is that the skeleton never fully hardens. The bones remain cartilaginous, soft and flexible, which turns out to be an advantage under extreme pressure. A rigid skeleton would be energetically expensive to maintain, and would provide no structural benefit in an environment where the water itself provides buoyancy and support. Cartilage bends where bone would crack. Genes linked to skin pigmentation were also absent. At depths where no light penetrates, there is nothing to see and nothing to be seen by. Coloration, whether for camouflage, mate attraction, or species recognition, serves no function. The metabolic cost of producing and maintaining pigment cells is a waste of energy that the organism cannot afford. And so the genes are lost and the skin becomes translucent, a pale ghostly membrane through which the muscles and organs are visible. Vision, too, has been abandoned. The genes that code for the light-sensitive proteins in the retina, the opsins that allow eyes to detect photons and translate them into electrical signals, are degraded or missing. Some deep-sea species retain vestigial eyes, small and non-functional, remnants of an ancestor that once lived in the light. Others have lost them entirely. In an environment of total darkness, an eye is not an asset. It is a liability. It is a complex organ that requires energy to build, maintain, and operate, and it returns nothing. Evolution, operating under the merciless calculus of energy budgets at extreme depth, simply removes it. This is not the gentle passive loss that we associate with decay. It is active editing. The genome is being pruned. Functions that do not pay for themselves in terms of survival are stripped away, and the energy that would have been spent on them is redirected to the systems that matter. More TMAO production, more efficient lipid storage, better mechanisms for tolerating cold and pressure. the deep-sea organism does not become less alive. It becomes more precisely alive, calibrated to a specific set of conditions with no room for redundancy. Consider the deep-sea dragonfish, one of the more dramatic examples of what evolution under darkness can produce. Its teeth are transparent. Under electron microscope examination, researchers have found that they are formed from nanocrystalline hydroxyapatite, a mineral component of bone, but arranged in a structure that is closer to natural glass than to anything we typically associate with teeth. The crystals are organized in a way that prevents light from scattering, rendering the teeth nearly invisible even under illumination. In the lightless depths where the dragonfish hunts, this means that its open jaws are effectively undetectable to prey. The jaw itself can open to nearly 90 degrees, a hinge mechanism that allows the dragonfish to engulf organisms that are a significant fraction of its own body size. The body measures roughly 25 centimeters, but nearly every structure on that body is specialized for a single purpose. The bioluminescent barbel that hangs from its chin, used to lure prey. The photophores along its flanks, used for communication or camouflage against the faint downwelling light that exists at shallower depths within its range. The expandable stomach that can accommodate meals larger than the fish itself, because in the deep sea you do not know when the next meal will come. Every feature serves a function. Nothing is decorative. Nothing is superfluous. In this environment, form is reduced to function, and function is reduced to survival. The reduction extends to the most fundamental processes of life itself. Many deep-sea species operate at metabolic rates that would be considered pathological in their shallow water relatives. Some function at roughly 10% of the metabolic rate of comparable species near the surface. Their hearts beat slower. Their digestion proceeds at a fraction of the normal speed. Their growth rates are measured in years where surface species would measure in months. This is not illness, it is economy. In an environment where food arrives unpredictably, sometimes separated by weeks or months, a fast metabolism is a death sentence. The organism that burns energy quickly will starve, long before the next meal drifts down from above. In the Haddle snailfish, this metabolic shift has a specific genetic signature, a gene called GPR27, which in surface-dwelling fish is involved in regulating metabolic rate and energy expenditure, has become inactive. The consequence is a shift in metabolic strategy. Instead of relying on rapid glucose metabolism, the Haddle snailfish tilts toward lipid storage and lipid utilization, a slower, more efficient way to extract energy from limited food resources. It can fast for extended periods without significant physiological cost, because its entire metabolic machinery is tuned for conservation, rather than expenditure. Day and night lose their influence at these depths. On the surface, virtually all life is organized around the 24-hour cycle of light and darkness. Circadian rhythms govern sleep, feeding, migration, reproduction, and hormone release. Even organisms that live in perpetual darkness, such as cave-dwelling fish, often retain vestiges of circadian biology, echoes of an ancestral life in the light. But in the Haddle zone, where no sunlight has ever reached, the very concept of a day becomes meaningless. There is no dawn. There is no dusk. There is only the slow, constant darkness and the occasional arrival of organic matter falling from the world above, what marine scientists call marine snow. A gentle perpetual rain of dead plankton, fecal pellets, and the remains of organisms that lived and died thousands of meters overhead. Some of the creatures that live at Haddle depths have existed in forms similar to their current ones for tens of millions of years. The amphipods, those crustacean scavengers that congregate in swarms around bait at the bottom of the Mariana Trench, belong to lineages that have occupied the deep sea since before the trench itself existed. Their body plan, a flattened, segmented form with multiple pairs of legs and a pair of large compound eyes that in many Haddle species have been reduced to near uselessness, has proven so effective at this depth that evolution has had little reason to alter it. They eat what falls, they reproduce in the dark, they persist. And then there is the phenomenon known as deep-sea gigantism, which seems at first to contradict the principle of reduction but, on closer examination, confirms it. Certain deep-sea organisms grow to sizes far exceeding their shallow-water counterparts. The supergiant amphipod, Alicella gigantea, can reach 34 centimetres, a startling size for a creature whose relatives near the surface rarely exceed 1 or 2 centimetres. Giant isopods, deep-sea cousins of terrestrial woodlice, can grow to 50 centimetres. The mechanisms behind this gigantism are debated, but the leading hypotheses involve the combination of cold temperatures, which slow metabolism and extend lifespan, and the low predation pressure at extreme depth. Without predators to keep them small and fast, these organisms can grow slowly and continuously over decades. Size in this context is not complexity, it is patience. The xenophyophores are perhaps the most unsettling expression of a hadal life. They are protists, single-celled organisms, but they build elaborate external structures called tests from the sediment and debris that settles on the ocean floor. These tests can reach 20 centimetres in diameter, making xenophyophores among the largest single-celled organisms on Earth. They sit on the seafloor of the Mariana Trench, immobile, filter-feeding from the water and the sediment around them, their tests accumulating barium and other heavy metals from their environment. A single cell the size of a small grapefruit, living at the deepest point on the planet, neither moving nor needing to move, having arrived at a form so minimal that it cannot be reduced further. What the Mariana Trench teaches us about evolution is uncomfortable precisely because it contradicts the narrative we prefer. We like to think of evolution as progress, as a march from simplicity to complexity, from lesser to greater, from primitive to advanced. The history of life on the surface supports this narrative in broad outline, and it is a narrative that places us, with our large brains and intricate social structures, at the leading edge. But in the trench, evolution has no such direction. It moves toward whatever works, and at 11 kilometres of depth, what works is less bone, less colour, less sight, less speed, less metabolism, less of almost everything except the chemistry required to survive. The organisms here did not lose their way, they found another one. It is worth sitting with that for a moment because the implications reach beyond biology. The trench does not reward ambition. It does not select for speed or strength or intelligence. It selects for endurance, for efficiency, for the ability to persist on almost nothing in a place that offers almost nothing. The creatures here are not failures of evolution. They are its quietest, most patient successes. And they have been succeeding in silence for millions of years. If the Mariana Trench has altered our understanding of form and speed, then deeper still, it challenges something more fundamental. It challenges the definition of life itself, or rather, it challenges the assumptions we have built the definition upon. For centuries, biology has treated sunlight as the foundation of nearly all ecosystems on Earth. The logic is simple and, on the surface, appears unassailable. Photosynthesis captures the energy of the sun. Plants and algae and cyanobacteria use that energy to convert carbon dioxide and water into sugars. Those sugars feed herbivores. Herbivores feed carnivores. Carnivores die and decompose, returning nutrients to the soil and the water, where the cycle begins again. It is a chain, and at the bottom of that chain, holding everything else up, is sunlight. When scientists first began to explore the deep ocean in the 19th century, they expected to find very little life beyond the reach of the sun. The prevailing theory, championed by the naturalist Edward Forbes in the 1840s, was that life in the ocean diminished with depth and ceased entirely below about 550 metres, a boundary he called the Azoic zone. Life, Forbes argued, needed light. Without light there could be no photosynthesis. Without photosynthesis there could be no food, and without food there could be no life. Forbes was wrong. His theory collapsed within decades as trawling expeditions brought up organisms from depth far exceeding his proposed limit. The HMS Challenger expedition of 1872 to 1876 dredged living creatures from every depth it sampled, including organisms from the floor of the Mariana Trench itself. Life, it turned out, did not end where the light ended. It continued. But the question of how it continued, of where the energy came from, would take another century to answer. The answer arrived in 1977, in a place called the Galapagos Rift, where geologists aboard the research submersible Alvin descended to explore a volcanic spreading centre on the Pacific Ocean floor. What they found there changed biology permanently. Clustered around jets of superheated mineral-laden water, pouring from cracks in the ocean floor were thriving communities of organisms. Giant tube worms, nearly 2 meters long with no mouth, no gut, and no anus. Mussels, clams, shrimp, an entire ecosystem flourishing in the total darkness, at depths where no sunlight had ever reached. The energy source was not the sun, it was the earth itself. The process is called chemosynthesis. Instead of capturing energy from light, as photosynthetic organisms do, chemosynthetic bacteria and archaea extract energy from chemical reactions. They oxidize compounds such as hydrogen sulfide, hydrogen gas, and methane, all of which are released by geological processes deep within the planet. The energy liberated by these reactions is used to fix carbon, to build organic molecules to sustain life. It is photosynthesis without the photo. Life powered not by a star 150 million kilometers away, but by the heat and chemistry of the rock beneath the ocean floor. At the bottom of the Mariana Trench, chemosynthesis operates through a specific geological mechanism called serpentinization. The process begins when seawater seeps into fractures in the ocean floor and comes into contact with a type of rock called peridotite, which is rich in the mineral olivine. Olivine is an iron-magnesium silicate, a common component of the Earth's upper mantle. When olivine reacts with water, it undergoes a chemical transformation. The iron in the olivine oxidizes, stripping electrons from hydrogen atoms in the water and releasing molecular hydrogen gas. At the same time, carbon dioxide dissolved in the seawater is reduced to methane. The reaction is exothermic, meaning it releases heat. The products, hydrogen, methane, and heat, are exactly what chemosynthetic microorganisms need to survive. The rock that results from this reaction is called serpentinite, named for its greenish scaly appearance, reminiscent of a serpent's skin. Serpentinite has been found throughout the Mariana Trench system, particularly along the forearc, the region between the trench and the volcanic island arc to the west. Serpentinite mud volcanoes, structures formed by the upwelling of serpentinized material from deep within the subduction zone, have been documented at multiple locations along the Mariana forearc. These mud volcanoes release highly alkaline fluids rich in hydrogen and methane, and they have been shown to sustain chemosynthetic communities of bacteria, mussels and clams at depths of over 4,000 meters. But the discovery that changed our understanding of the Mariana Trench specifically came in 2012 during an expedition led by James Cameron aboard the Deep-Sea Challenger. Kevin Hand, an astrobiologist at NASA's Jet Propulsion Laboratory, was part of the scientific team. Using a lander equipped with cameras, sampling bottles and a depth sensor, the team deployed instruments to the inner slope of the Mariana Trench at a depth of over 10,600 meters. When they reviewed the images captured by the lander, they saw something unexpected. Greenish, droopy, finger-like filaments clinging to exposed rock. The structures were consistent in appearance with microbial mats, dense communities of bacteria and archaea that grow on surfaces where chemical energy is available. Analysis of the sediment and rock samples retrieved from the site confirmed the presence of serpentinized peridotite. The chemical signatures in the water samples were consistent with active serpentinization, meaning that hydrogen and methane were being produced at or near the site. The filaments, if they were indeed microbial in origin, would represent the deepest known chemosynthetic community on Earth, living at a depth of more than 10,600 meters, sustained entirely by energy produced by reactions between rock and water. Ham was careful to note that the discovery needed confirmation. The team had not been able to physically retrieve a sample of the filamentous material itself. The identification was based on images and on the chemical context of the surrounding rock and water, but the evidence was compelling, and the implications were profound. In a separate but related finding, researchers in 2013 reported the discovery of bacteria living not on the ocean floor, but within the oceanic crust itself. Beneath layers of sediment embedded in the basaltic rock of the seafloor, microorganisms were found to be metabolizing hydrogen produced by reactions between the rock and seawater. These organisms were entirely separate from the hydrothermal vent systems. They did not depend on the concentrated chemical energy of a black smoker or a white smoker. They were sustained instead by the slow, diffuse release of hydrogen from the rock itself. A biosphere within the rock, life inside the Earth's crust, powered by nothing more than the interaction of water and stone. What these discoveries suggest is that the deepest ecosystems on Earth operate according to principles that are fundamentally independent of the surface world. They do not need sunlight. They do not need photosynthesis. They do not need the atmosphere. They require only rock water and the chemistry that occurs when the two meet. Consider the scale of time involved. On the surface of the Earth, life has been catastrophically disrupted at least 5 times. The end Ordovician extinction, roughly 444 million years ago. The late Devonian extinction, around 372 million years ago. The end Permian extinction, approximately 252 million years ago, which killed an estimated 96% of all marine species. The end Triassic extinction, about 201 million years ago. And the end Cretaceous extinction, 66 million years ago, which ended the reign of the dinosaurs. Each of these events reshaped life on the surface. Ecosystems were destroyed and rebuilt. Lineages that had dominated for millions of years were wiped out in geological instants. The survivors radiated into the emptied niches, diversifying into new forms, building new food webs, starting again. But the chemosynthetic ecosystems deep below the Mariana Trench may have been insulated from all of it. Ice ages do not reach 11 kilometers of depth. Asteroid impacts, however devastating to the surface, do not significantly alter the temperature or chemistry of the deep ocean on the timescales that matter to these organisms. Atmospheric collapse, ozone depletion, volcanic winter, none of these events penetrate to the floor of the Mariana Trench in ways that would disrupt the slow, steady release of hydrogen from serpentinizing rock. If this is true, and the evidence increasingly suggests that it is, then the chemosynthetic communities at the bottom of the Mariana Trench may represent some of the most ancient, continuously operating ecosystems on the planet. Not the oldest individual organisms necessarily, but the oldest type of ecosystem, the oldest way of making a living. Some researchers have gone further. There is a growing body of evidence suggesting that chemosynthesis may have preceded photosynthesis as a form of metabolism on Earth. The earliest life, which appeared roughly 3.5 to 4 billion years ago, existed in an environment with very little free oxygen and with geological activity far more intense than what we see today. Hydrothermal vents and serpentinization reactions would have been abundant. Sunlight, while present, was not yet being harnessed by any organism in the way that modern cyanobacteria harness it. The chemical building blocks for chemosynthesis, hydrogen, methane, hydrogen sulfide, were available everywhere that rock met water. If chemosynthesis did come first, then the ecosystems at the bottom of the Mariana Trench may resemble, in their broad outlines, the first ecosystems that ever existed on Earth. Not identical, certainly. The specific organisms are different, the geology is different, but the principle, life sustained by the energy released when water reacts with rock, may be among the oldest principles of biology on this planet. older than photosynthesis, older than the ozone layer, older than free oxygen in the atmosphere, older perhaps than the surface world as we know it. And this is where the story begins to reach beyond Earth entirely. Serpentinization is not unique to our planet. It occurs wherever olivine meets water, and olivine is among the most common minerals in the solar system. The moons of the outer planets, Europa and Enceladus in particular, are now known to possess subsurface oceans of liquid water in contact with rocky cores. In 2005, the Cassini spacecraft observed plumes of water vapour and ice particles erupting from the south pole of Enceladus. Subsequent analysis revealed the presence of molecular hydrogen in those plumes, a signature consistent with serpentinization occurring at the moon's rocky seafloor. Europa, Jupiter's ice-covered moon, is thought to possess a global ocean beneath its frozen crust, an ocean that may contain 2 to 3 times as much water as all of Earth's oceans combined. And that ocean sits in contact with a rocky mantle, where serpentinization could, in principle, sustain chemosynthetic life. Kevin Hand, the same NASA astrobiologist who identified the possible microbial mats at Serena Deep in the Mariana Trench, has spent much of his career arguing that the search for extraterrestrial life should focus on these icy moons. The logic is direct. If life can exist at the bottom of the Mariana Trench, sustained entirely by serpentinization, with no sunlight, no photosynthesis, and no connection to the surface, then it could, in principle, exist at the bottom of Europa's ocean or Enceladus's subsurface sea. The conditions are not identical. The pressures, temperatures, and chemical environments differ. But the fundamental mechanism, rock plus water equals energy, is the same. The deepest place on Earth may be telling us something about the deepest places in the solar system, and the life that persists in the Mariana Trench, silent and ancient and indifferent to the sky above, may be the closest model we have for what we might one day find on another world. We came to the trench looking for the boundary of life. Instead, we may have found its origin. And that origin, it turns out, does not require a star. It requires only rock and water and time. And the Mariana Trench has had all three in abundance for 52 million years. The Mariana Trench is not only about the life that survives there, it is also about us. About what remains of the human mind when every familiar reference is removed. About what happens to the architecture of human perception when it is placed in a setting it was never built for. We do not think about it often, Moan, but the human brain constructs its sense of the world from a continuous stream of sensory data. Light entering the eyes tells us where objects are, how far away they are, how fast they are moving. Sound arriving at the ears provides spatial information, context warning. The feeling of gravity pulling on the body tells us which direction is down. Temperature variations across the skin tell us where heat sources are and where cold surfaces lie. Even the subtle pressure of air moving across the face contributes to our sense of being somewhere, of occupying a specific point in a specific space. Remove all of these inputs at once, and something disturbing happens. The brain does not simply go quiet. It does not simply wait in stillness for the signals to return. Instead, it begins to generate its own. The aphotic zone of the ocean begins at approximately 1,000 meters of depth. Below this line, the darkness is total, not dimmed, not reduced, total. No photon of sunlight has reached these depths in the history of the planet. But total darkness is only the beginning of what the deep ocean removes. Sound, which travels efficiently through water, is present, but in the deepest parts of the trench it is reduced to the low, distant rumble of geological movement, the creak of tectonic plates, and the faint hiss of sediment settling. There are no animal calls at these depths, no reef sounds, no breaking waves, nothing that the human auditory system can latch onto as a reference point. Temperature, too, ceases to provide orientation. In the Hadal zone, the water temperature holds steady between 1 and 4 degrees Celsius, with virtually no variation across space or time. On the surface we use temperature gradients constantly, without realizing it. The warmth of sunlight on one side of the face tells us which way we are facing. The cool draft from an open window tells us where the window is. In the trench, there is no gradient. The cold is uniform, featureless, and unrelenting. The result is not merely darkness. It is the loss of reference itself. The brain cannot determine where it is, how it is oriented, or how much time has passed. It cannot distinguish between looking up and looking down. It cannot estimate distance, because there are no visual cues from which to calculate it. The vestibular system, the inner ear mechanism that senses acceleration and gravity, still functions, but without visual confirmation, even its signals become unreliable. Astronauts in microgravity report similar experiences. Without a visual horizon to confirm which way is up, the inner ear's signals become ambiguous, and the brain's model of the body's position in space begins to drift. In 2009, a study conducted by Oliver Mason and Francesca Brady at University College London explored what happens to the human mind under conditions of sensory deprivation, far less extreme than those found in the deep ocean. They placed 19 healthy volunteers in a completely dark, soundproof chamber for 15 minutes. Just 15 minutes. Not hours, not days. 15 minutes of darkness and silence. The results were striking. Within that short period, several participants began to report visual hallucinations. They saw shapes, patterns, and faces that were not there. Some reported a sense of presence, the feeling that another person or entity was in the room with them, though they knew the room was empty. Others experienced distortions of time perception, with some feeling that far more or far less time had passed than actually had. A few reported heightened anxiety and a sense of losing their grip on their own identity, a dissolution of the boundary between self and surroundings. These were healthy volunteers. They were not sleep-deprived, not under the influence of any substance, not suffering from any psychiatric condition. They were simply placed in the dark and the quiet for a quarter of an hour, and their brains, deprived of the sensory anchors they depend on, began to fill the void. The phenomenon is well documented across extreme environments. Submarine crews on long deployments in the deep ocean have reported similar experiences. The enclosed, dimly lit, monotonous environment of a submarine, while not as extreme as the trench itself, shares some of its sensory characteristics. Studies of polar expeditions, where participants endure months of continuous darkness during the polar winter, have documented anxiety, irritability, depression, sleep disturbance, and in some cases what researchers call polar madness, or winter over syndrome, a cluster of psychological symptoms associated with prolonged sensory and social monotony. Cave explorers who spend days or weeks in total darkness underground report distortions of time perception so severe that their circadian rhythms, the internal biological clocks that govern sleep and wakefulness, can drift by hours or even reverse entirely. Without the external cue of sunlight to reset the clock, the body begins to follow its own idiosyncratic rhythm, and the subjective experience of time becomes increasingly disconnected from objective reality. What all of these environments share is not danger in the conventional sense. They are not places where the threat is a predator, or a falling rock, or a sudden storm. The threat is subtler and, in some ways, more fundamental. It is the gradual erosion of the sensory framework that the brain relies on to construct a coherent model of the world and of the self. Remove that framework, and the model begins to degrade. The boundaries between real and imagined, between self and environment, between inside and outside begin to soften. The Mariana Trench represents perhaps the most extreme version of this experience that the Earth can offer a human being. At nearly 11 kilometers of depth, in complete darkness, near freezing temperatures, a pressure of 1,100 atmospheres, and a silence broken only by the movement of rock, the human brain would be stripped of almost every input it depends on. The body would survive inside the titanium sphere of a deep-sea submersible. The mind would be less certain of its survival. In December of 2014, something happened in the Challenger Deep that illustrates, perhaps better than any experiment, the relationship between human technology and the deep. The Schmidt Ocean Institute had commissioned an expedition aboard the research vessel Falkor, led by Chief Scientist Douglas Bartlett of the Scripps Institution of Oceanography. One of the mission's objectives was to record for the first time the ambient sound at the bottom of the Mariana Trench, not the biological sounds, not the sounds of marine animals or geological events, the simple background noise of the deepest place on Earth, the sound of the deep itself. To accomplish this, the team deployed two instruments. They were called Deep Sound 2 and Deep Sound 3, and they were free-falling acoustic profilers designed to sink through the water column under their own weight, recording sound as they descended. Each instrument was housed inside a glass sphere, a hollow ball of specialized glass roughly 15 inches in diameter, chosen because glass distributes pressure evenly and can withstand enormous forces if manufactured without imperfections. The instruments were designed to descend to the floor of Challenger Deep, roughly 10,900 meters, and then release their ballast weights and float back to the surface, carrying their recordings with them. Both instruments were deployed on December 16th, 2014. They sank through the water, recording the ambient sound of the ocean as they fell. At approximately 9,000 meters, both instruments were recording normally. The sound they captured was quiet, remarkably quiet. The deep ocean, far from the surface storms and biological activity of shallower waters, is one of the quietest places on Earth. The recordings showed a low, featureless hum, the combined effect of distant earthquakes, ocean currents, and the thermal noise of water molecules in random motion. And then, at a depth of approximately 8,620 meters, something went wrong. The glass sphere housing Deep Sound 3 failed. It imploded, collapsing inward as the pressure of the surrounding water overwhelmed its structural integrity. The implosion was instantaneous and violent, converting the glass housing and the air it contained into a shock wave that propagated outward through the water in every direction. Deep Sound 2, which was still descending nearby, recorded the entire event. The recording begins with the sharp crack of the initial failure, a sound like a gunshot amplified and distorted by the medium of water. Then comes the shock wave, a pulse of compressed water that expands outward from the point of implosion. And then, remarkably, come the echoes. The shock wave bounced off the floor of the Challenger Deep below and the surface of the ocean above, travelling back and forth between them in a series of reflections that lasted for more than a minute. The Challenger Deep, that narrow, uneven valley at the bottom of the ocean, rang like a bell struck in a cathedral. The sound reverberated through the trench, each echo slightly fainter than the last, until it faded into the ambient silence of the deep. David Barclay, the scientist who had designed the deep sound instruments, was aboard the Falkor when it happened. He heard the initial pop through an underwater microphone connected to the ship's hull. He described the moment with the understated precision of a scientist who has just watched his instrument die. The frustration of losing Deep Sound 3, he wrote later, gradually gave way to morbid curiosity about the data it had inadvertently produced, and that data turned out to be extraordinary. By analysing the timing of the echoes, the intervals between each reflection of the shock wave off the seafloor and the surface, the team was able to calculate the depth of the Challenger Deep with remarkable precision. They accounted for variations in the speed of sound at different depths, which changes with temperature, salinity, and pressure, and they arrived at a figure, 10,983 meters, plus or minus 6 meters. It is, to this day, among the deepest and most precisely constrained estimates of the depth of the Challenger Deep ever produced. There is something almost poetic in the fact that the only sound we have ever recorded from the very deepest part of the ocean is the sound of our own technology failing. We sent an instrument to listen to the silence, and the silence destroyed it. And in its destruction, it told us something we could not otherwise have measured. The recording exists. You can listen to it. It is publicly available through the Schmidt Ocean Institute. It begins with silence, the ambient noise of the deep, then the crack, then the reverberations fading, until the trench is quiet again. It is not a long recording, but it is one of the most singular pieces of audio in the history of science. The voice of the deepest place on Earth, singing back to us the sound of our own failure. And perhaps that is the real lesson of the abyss. The Mariana Trench does not rely on monsters or darkness or crushing pressure to be unsettling. Those are the elements of fiction. The reality is quieter and more unsettling. The trench is a place that removes. It removes light. It removes sound. It removes temperature variation. It removes every sensory anchor that the human brain depends on to maintain its model of itself and its world. And in the absence of those anchors, the mind begins to dissolve, not dramatically, not violently, gently, like a structure built on sand, slowly being reclaimed by the tide. The threat of the deep is not death. Death at least is an event. The deep threatens something subtler, the gradual unmaking of orientation, the dissolution of certainty about where you are, which way you face, how much time has passed, whether the shapes you see in the darkness are real or manufactured by a brain desperate for input. Down there, people do not die of the dark. They begin to lose their sense of themselves, and that perhaps is more frightening than any creature the trench could contain. The darkness does not need to be filled with terrors. It only needs to be empty. The brain will supply the rest. There is a fact about the Mariana Trench that is so simple it tends to be overlooked. After more than 150 years of measurement, after dozens of expeditions and the deployment of technologies ranging from weighted ropes to nuclear-powered submarines to satellite-calibrated sonar arrays, we still do not know exactly how deep it is. Not approximately. We know it approximately. The number sits somewhere in the range of 10,900 to 10,990 meters, depending on which study you consult, which instrument was used, and which assumptions were made about the speed of sound in the water column at the time of measurement. But the exact depth, the number that would satisfy a physicist or a surveyor, remains elusive. The deepest point on Earth carries a margin of uncertainty measured in tens of meters, and that margin, far from narrowing over time, has in some ways grown more complex as the methods of measurement have become more sophisticated. The history of attempting to measure the Challenger deep reads like a case study in the difficulty of knowing anything precisely about a place that actively resists our instruments. The first sounding of the Mariana Trench was made in 1875, during the voyage of HMS Challenger, the British naval corvette that had been repurposed for the world's first systematic oceanographic expedition. The Challenger's method of measuring depth was brutally simple. A weight was attached to a length of hemp rope and lowered over the side until it reached the bottom. The length of rope paid out was measured, and that measurement became the depth. On March the 23rd, 1875, in the waters east of the Mariana Islands, the Challenger's sounding weight touched bottom at a depth of 4,475 fathoms, or roughly 8,184 meters. It was, at the time, the deepest sounding ever recorded. The measurement was crude by modern standards, affected by currents that could have carried the rope sideways during its descent, by the weight of the rope itself, and by the difficulty of knowing the precise moment when the weight reached the bottom. But it was enough to establish that the Mariana Trench contained water of extraordinary depth. The name Challenger Deep was formalized when the second ship to bear that name, HMS Challenger II, returned to the area in 1951 during a hydrographic survey. Using an echo sounder, a device that measures depth by transmitting a pulse of sound and timing its return, the survey recorded a maximum depth of approximately 10,863 meters. This was nearly 3 kilometers deeper than the original 1875 sounding, a discrepancy explained by the limitations of the rope and weight method and by the fact that the 1875 sounding had been taken at a different location within the trench. In 1957, the Soviet research vessel Vityaz passed through the Mariana Trench on a transit between research stations and recorded a depth of 11,034 meters, plus or minus 50 meters. This measurement, taken with a single beam echo sounder, was for decades cited as the deepest depth in the ocean, but it carried a significant margin of error, and subsequent measurements would not confirm it. On January 23, 1960, the Bathyscaphe Trieste descended to the floor of the Challenger Deep, carrying two people, Jacques Piccard and Don Walsh. It was the first time a human being had reached the bottom of the ocean's deepest point. The instruments aboard the Trieste initially indicated a depth of 11,521 meters, but this was later revised to 10,916 meters after a recalculation. The revision highlights a recurring problem in deep ocean measurement. The instruments aboard the Trieste measured pressure, not depth directly. Converting pressure to depth requires knowing the precise density of the water at every point along the descent, which in turn requires knowing the temperature, salinity, and pressure at each point. Any error in these environmental profiles propagates into the final depth calculation. In 2012, filmmaker and explorer James Cameron made the third crewed descent to the Challenger Deep, piloting the submersible Deep-Sea Challenger on a solo dive. His depth was recorded at approximately 10,908 meters. In 2019, Victor Vescovo descended to 10,927 meters aboard the submersible limiting factor as part of his 5 Deeps expedition, a mission to reach the deepest point in each of the world's 5 oceans. His team made 5 separate dives to the Challenger Deep, and their measurements, derived from calibrated pressure gauges, cluster tightly around 10,925 meters for the deepest point. In 2021, the accidental implosion of Deep Sound 3 provided yet another estimate. As we discussed, the timing of the shockwave echoes, analysed by Scott Loringer and his colleagues, yielded a depth of 10,983 meters, plus or minus 6 meters. This is among the deepest estimates ever published, and it sits notably above the cluster of measurements from the manned submersibles. Also in 2021, a comprehensive analysis by Hans van Haren and colleagues, using data from multiple sonar surveys, reported a depth of approximately 10,935 meters. They noted that earlier multi-beam sonar surveys, including an influential 2010 survey by Gardner and colleagues, had overestimated the depth by roughly 69 meters due to the use of a sound velocity profile that was not sufficiently calibrated to the conditions at the time of measurement. And so the numbers accumulate: 10,916, 10,927, 10,935, 10,983, each measured with different instruments at slightly different locations within the Challenger Deep, under slightly different environmental conditions, and each carrying its own margin of error. Over 20 estimates have been published in 150 years. The numbers shift with each new measurement, not because the trench is changing, although it is, as we shall see, but because the act of measurement in this environment is fundamentally constrained by the physics of the medium. The central problem is sound. Nearly all modern depth measurements in the deep ocean rely on acoustics, on the transmission and reception of sound waves. A sonar system emits a pulse of sound. That pulse travels downward through the water, reflects off the seafloor, and returns to the receiver. The depth is calculated from the time it takes the sound to make the round trip, divided by 2 and multiplied by the speed of sound in the water. But the speed of sound in water is not constant. It varies with temperature and salinity and pressure. In the upper ocean, where temperatures change rapidly with depth, the speed of sound can vary by tens of meters per second over short vertical distances. In the deep ocean, Where temperatures are more stable, pressure becomes the dominant factor, and the speed of sound increases steadily with depth. A sound pulse travelling from the surface to the bottom of the Challenger. Deep passes through water, whose acoustic properties change continuously along the way. To calculate depth accurately, you need a sound velocity profile, a detailed record of how the speed of sound varies with depth at the specific location and time of the measurement. Any error in that profile translates directly into an error in the calculated depth. Taking an accurate sound velocity profile in the Challenger Deep requires lowering instruments to within meters of the bottom, a procedure that is expensive, time-consuming and technically challenging. Some surveys have used profiles taken kilometers away from the actual measurement site, introducing spatial errors. Others have used profiles from different seasons, when water temperatures may have differed. The differences are small, fractions of a meter per second in sound speed, but over a sound path of nearly 22 kilometers, the round trip from surface to trench floor and back, those fractions compound. There is also the matter of geometry. The Challenger Deep is not a flat plain. It is a narrow, uneven valley, roughly 11 kilometers long and a few kilometers wide, with steep walls and an irregular floor. The deepest point may be a small depression within this larger feature, perhaps only a few hundred meters across. A sonar beam fired from a ship at the surface spreads as it descends, and by the time it reaches the bottom, it may illuminate an area hundreds of meters wide. The depth reported is the depth of the shallowest point within that footprint, not necessarily the deepest point within it. To find the true deepest point, you would need to map the floor of the Challenger Deep at a resolution of meters, a task that requires either multiple passes with a narrow beam sonar or a close-range survey from a submersible. Even submersible-based measurements, which use pressure gauges rather than acoustics, are not immune to uncertainty. Pressure gauges must be calibrated, and the conversion from pressure to depth still requires knowledge of the water column's density profile. The difference between a calibration performed at the surface and one performed at depth can introduce errors of several meters. All of this means that the deepest point on Earth, a specific spot on the floor of the Challenger Deep, is known to within roughly 50 to 80 meters of its true depth. We know it is deeper than 10,900 meters and almost certainly shallower than 11,000 meters. But within that window, the exact number remains a subject of ongoing measurement and debate. It is often said that only 5% of the ocean floor has been explored in any detail. This figure, while commonly cited, is difficult to verify precisely, but it captures a genuine truth. The ocean covers roughly 361 million square kilometers of the Earth's surface, and the vast majority of it has never been surveyed, with the kind of high-resolution sonar that would reveal features smaller than a few hundred meters across. In many parts of the deep ocean, the best available maps have a resolution of 1 to 5 kilometers, meaning that features the size of a city block would be invisible. We have more detailed maps of the surface of Mars, which has been surveyed by orbiting spacecraft with radar instruments, than we do of the floor of our own ocean. The limitation is not curiosity. Scientists would dearly love to map every square meter of the deep ocean. The limitation is logistics. A research vessel conducting a high-resolution sonar survey can cover at best a few hundred square kilometers per day. At that rate, mapping the entire ocean floor would take thousands of ship years. The cost would be measured in billions of dollars, and the data, once collected, would require years of processing and interpretation. More people have walked on the surface of the moon than have descended to the Challenger Deep. 12 astronauts walked on the moon between 1969 and 1972. As of 2025, fewer than 30 people have descended to the deepest point in the ocean, including the crews of Trieste, Deep Sea Challenger, and the various dives of the limiting factor. The moon is 384,000 kilometers away. The Challenger Deep is 11 kilometers below the surface of the sea. The distance is incomparable, and yet the deep ocean remains less visited, less mapped, and less understood than the surface of another world. There is something humbling about this. We have sent robots to Mars, probes to the outer planets, telescopes to the edge of the observable universe. We have photographed the shadow of a black hole 55 million light years away. And we cannot agree to the nearest 50 meters on the depth of the deepest place on our own planet. The trench does not hide its depth from us deliberately. It is not hostile. It is simply indifferent. The physics of sound propagation, the geometry of a narrow valley, the variability of temperature and salinity in the water column, these are not obstacles placed in our way. They are the natural properties of the environment, but they are sufficient to ensure that the deepest point on Earth remains, after a century and a half of effort, imperfectly known. We have measured this place more than 20 times, and we still cannot agree on how deep it is. Perhaps that more than anything reveals the nature of the relationship between human knowledge and the deep sea. We do not study the trench from a position of mastery. We approach it, instrument in hand, and the instrument returns an answer that is close, but never final. We refine our methods, improve our technology, account for more sources of error, and the answer shifts slightly, settles into a narrower range, but never resolves into a single definitive number. The trench is patient with our efforts. It neither cooperates nor resists. It simply waits, 11 kilometers down, for the next measurement. And the next measurement will be, like all the others, an approximation. Even if we could measure the Mariana Trench perfectly, even if we could pin down the depth of the Challenger Deep to the nearest centimetre, the number would not stay the same. Most people picture the Mariana Trench as a fixed feature of the Earth's surface, a crack in the ocean floor that has always been there and always will be, a geological fact, stable and unchanging like a mountain or a river canyon. But the trench is not a static feature. It is a process. It is the visible expression of one of the most powerful forces operating on the surface of this planet, a force that has been reshaping the Earth's crust for billions of years and that continues right now, at this moment, as you listen to these words. The Mariana Trench exists because the Pacific Plate, one of the largest tectonic plates on Earth, is diving beneath the Philippine Sea Plate in a process called subduction. The Pacific Plate is oceanic crust, dense and heavy, formed at a mid-ocean ridge millions of years ago, and slowly moving westward across the Pacific Basin. The Philippine Sea Plate is also oceanic crust, but it overlies the descending Pacific Plate, and the boundary between the two forms the trench. At a subduction zone, one plate descends beneath another and sinks into the Earth's mantle. The trench is the surface expression of this descent, the point at which the leading edge of the sinking plate bends downward and begins its journey into the interior of the planet. The depth of the trench is a function of the angle and speed of that descent, the age and density of the descending plate, and the geometry of the overlying plate. Near Guam, the Pacific Plate converges with the Philippine Sea Plate at a rate of roughly 60 to 70 millimeters per year. This is not fast by any human standard. It is less than the rate at which your fingernails grow. It is roughly the speed at which a glacier moves. At any given moment, the motion is imperceptible. You could stand on either plate and never feel a thing, but sustained over geological time, this motion is immense. The subduction at the Mariana Trench began approximately 52 million years ago, during the Eocene epoch, when the Pacific Plate first began to descend beneath the edge of the Philippine Sea Plate. In the time since, at 60 to 70 millimeters per year, the Pacific Plate has travelled more than 3,000 kilometers into the Earth's mantle, 3,000 kilometers of oceanic crust, consumed by the interior of the planet, 1 millimeter at a time. That steady, patient motion has produced the deepest feature on the surface of the Earth. The process is ongoing. Right now, at this moment, the Pacific Plate is sliding beneath the Philippine Sea Plate. The trench is being actively maintained by this motion. If subduction were to stop, which it will not for tens of millions of years at least, the trench would gradually fill with sediment and disappear. It exists only because the process that creates it continues. The Mariana subduction zone is unusual in several respects. In most subduction zones around the world, the descending plate dips at a moderate angle, typically between 30 and 45 degrees. The angle determines many of the features associated with the subduction zone, including the width of the volcanic arc behind the trench, the pattern of earthquakes, and the depth of the trench itself. At the Mariana Trench, however, the Pacific Plate plunges at a much steeper angle, in some places approaching nearly vertical. This steep descent is one of the reasons the trench is so deep. A plate descending at a steep angle creates a sharper bend at the surface and a deeper trench. Why the Pacific Plate descends so steeply at this location is not entirely understood. One leading hypothesis relates to the extreme age of the Pacific Plate in this region. The crust that is entering the trench near the Challenger Deep was formed at a mid-ocean ridge roughly 150 to 170 million years ago during the Jurassic period. Over that time it has cooled, become denser, and thickened. Old cold oceanic crust is significantly denser than the underlying mantle, and this density difference provides the driving force for subduction. The older and colder the crust, the more readily it sinks, and the steeper its angle of descent. The Pacific Plate at the Mariana Trench is among the oldest oceanic crust on the planet, and its density is correspondingly high. It does not merely subduct, it plunges. A second anomaly of the Mariana subduction zone is seismic. The Mariana Trench forms the largest subduction system on Earth. It is a feature of planetary scale, extending for thousands of kilometers across the western Pacific. And yet, unlike most of the world's other major subduction zones, it has not produced a giant earthquake in recorded history. This is remarkable because subduction zones are, globally, the most seismically active features on Earth. The largest earthquakes ever recorded have occurred at subduction zones. The 1960 Chilean earthquake, magnitude 9.5, the most powerful earthquake in the instrumental record, occurred at the boundary where the Nazca Plate subducts beneath the South American Plate. The 2004 Indian Ocean earthquake, magnitude 9.1, occurred at the Sunda subduction zone. The 2011 Tohoku earthquake, magnitude 9.0, occurred at the Japan Trench. These are events of extraordinary violence, releasing energy equivalent to billions of tons of explosives, generating tsunamis that travel across entire ocean basins and killing hundreds of thousands of people. The Mariana subduction zone, despite being comparable in scale to these other systems, has produced nothing remotely similar during the period for which we have reliable seismic records, roughly the last 150 years. The largest earthquake recorded at the Mariana Trench was approximately magnitude 7.7, a significant event by any standard, but orders of magnitude smaller than the great subduction earthquakes of Chile, Indonesia, and Japan. One possible explanation involves serpentinite, the mineral we encountered earlier in the context of chemosynthesis. Serpentinite is formed when seawater reacts with the olivine in mantle rock, a process that occurs extensively in the Mariana subduction zone. Serpentinite has a distinctive physical property. It is soft, significantly softer and more ductile than the unaltered rock from which it forms. When serpentinite is present along the boundary between two tectonic plates, it may act as a lubricant, reducing the friction between the plates and allowing them to slide past one another smoothly, rather than locking and then rupturing in a catastrophic earthquake. Seismic studies have detected a layer of serpentinized material in the upper mantle beneath the Mariana Trench, estimated at 4 to 6.5 kilometers in thickness. This layer sits at the boundary between the descending Pacific Plate and the overlying mantle wedge, exactly where the greatest frictional forces would otherwise be concentrated. If this serpentinite layer is sufficiently thick and continuous, it could explain the absence of great earthquakes at the Mariana Trench. The plates are not locked. They are gliding past one another on a layer of soft, slippery rock. But there is another possibility, and it is less reassuring. Jian Lin, a geophysicist at the Woods Hole Oceanographic Institution, has studied the seismicity of the Mariana subduction zone extensively. He has suggested that the absence of great earthquakes in the recorded history of the Mariana Trench may not mean that such earthquakes cannot occur. It may mean that they occur on timescales longer than our records can capture. The written seismic history of the Pacific region extends back a few centuries at most. In the Mariana Islands, reliable records go back only about 150 years. The instrumental record, the period during which earthquakes have been measured with calibrated seismographs, goes back about a century. These are, in geological terms, vanishingly short periods. A subduction zone that produces a great earthquake every 500 years, or every 2,000 years, would appear entirely quiet during a 150-year observation window. The Cascadia subduction zone off the coast of the Pacific Northwest provides a sobering precedent. For decades, this zone was considered relatively quiet, with no great earthquakes in the historical record of the region. It was not until geological research in the 1980s and 1990s revealed evidence of massive earthquakes and tsunamis in the deeper past, including a magnitude 9.0 event in the year 1700, that the Cascadia zone was recognized as capable of producing catastrophic seismic events. The absence of evidence had been mistaken for evidence of absence. Whether the Mariana Trench is genuinely aseismic, its plates gliding smoothly on serpentinite, or whether it is quietly accumulating strain that will one day be released in an event larger than any in the historical record, is a question that cannot be answered with the data we currently have. Both hypotheses are consistent with the available evidence, and the honest answer, the answer that a careful scientist would give, is that we do not know. The trench moves. It has been moving for 52 million years. It has consumed thousands of kilometers of oceanic crust, generated the deepest point on the Earth's surface, and created a geological environment unlike any other on the planet. And we have been watching it for barely a century and a half, a fraction of a fraction of a fraction of its history. There is a metaphor here, though it is not a comfortable one. We stand at the edge of the Mariana Trench with our instruments and our models and our 150 years of data, and we attempt to understand a process that operates on timescales of tens of millions of years. It is as if someone were to watch a single frame of a film that runs for a century and attempt to deduce the plot. We see a snapshot. The trench sees everything. And the trench for its part continues. The Pacific Plate descends. The trench deepens, fractions of a millimeter at a time. Sediment accumulates. Serpentinite lubricates the fault zone, or does not. And somewhere in the future, the plates will do what plates do, which is whatever the physics of the mantle dictates, regardless of whether we are watching or whether we understand. The trench does not move for us. It moves because it must. It has been doing so since before the first hominid stood upright, and it will continue long after the last one lies down. There is a kind of peace in that. The trench is beyond our timescale. It operates on a clock that measures in millions of years, a clock so slow that an entire human civilization fits between two of its ticks. Whatever happens to us, the trench will continue. The plates will keep moving. The process will go on. And that process, slow and silent and utterly indifferent to the human story unfolding on the surface above, is one of the most powerful forces on Earth. There is one detail about life in the deep ocean that often surprises people when they first encounter it. In a world of total darkness, where no sunlight has ever reached, light exists. Not borrowed light, not reflected light. Light that is manufactured by living organisms, molecule by molecule, reaction by reaction, in the dark. Bioluminescence, the production of light through chemical processes within a living body, is the most common form of communication in the deep ocean. Roughly 76% of all deep-sea organisms are estimated to produce their own light in some form. They use it to attract prey, to confuse predators, to signal to potential mates, and in some cases for reasons that scientists have not yet determined. The light is typically produced by a reaction between a molecule called luciferin and an enzyme called luciferase, and it is extraordinarily efficient. Nearly all of the energy involved in the reaction is converted to light, with almost none wasted as heat. By comparison, an incandescent light bulb converts roughly 5% of its electrical energy to light and the rest to heat. Bioluminescence is, in thermodynamic terms, one of the most efficient light sources known. In the Hadal zone, bioluminescence is less common than in the waters above, but it has not disappeared entirely. Some deep-sea bacteria produce faint, steady glows. Certain amphipod species carry bioluminescent bacteria in specialized organs. The light they produce is faint, barely detectable, a cold blue-green shimmer in water that has not seen the sun in the entire history of the Earth. It is life, making its own light, in the deepest dark the planet has to offer. Before we turn to the marks that human civilization has left on the trench, there is one more aspect of life at Hadal depths that deserves attention, because it defies one of our most basic assumptions about how ecosystems work. On the surface, food chains are relatively short and well understood. Sunlight feeds plants, plants feed herbivores, herbivores feed predators, waste and death return nutrients to the soil. The cycle repeats. At each level, energy is lost, roughly 90% per transfer, which is why there are always fewer predators than prey, fewer lions than zebras, fewer hawks than sparrows. This principle, the pyramid of energy, is one of the foundational concepts of ecology. In the Mariana Trench, the pyramid is inverted. At the very bottom, there are no plants. There is no photosynthesis. There are no herbivores in the traditional sense. The primary energy input comes from two sources. The first is marine snow, the continuous slow rain of organic particles falling from the surface ocean, thousands of meters above. These particles include dead plankton, fecal pellets, shed mucus, and the remains of organisms that lived and died in the sunlit zone. By the time they reach the Hadal zone, much of their nutritional value has been consumed by organisms at intermediate depths. What arrives at the bottom is often degraded, nutrient-poor, and sparse, yet it is the main food source for most Hadal organisms. The second source is the chemosynthetic production we discussed earlier, the energy derived from serpentinization and other geological processes. This energy supports bacterial and archaeal communities in the sediment and on exposed rock, and those communities in turn support a limited number of organisms that graze on them. What is striking about the Hadal food web is its structure. At the base are the bacteria and archaea. Above them are the amphipods, the dominant multicellular consumers at Hadal depths, scavengers that converge on any food source that reaches the bottom. And above the amphipods, at the top of the Hadal food chain, sits the snailfish, the translucent soft-boned creature we met earlier. The snailfish feeds primarily on amphipods. It is, in this environment, the apex predator. Consider what that means. The top predator, at the deepest point on Earth, is a fish the size of a human hand, translucent, boneless, blind or nearly so, drifting slowly through water at a temperature barely above freezing. It does not hunt in the way we typically imagine predators hunting. There is no chase. There is no ambush. The snailfish moves through the dark, sensing vibrations in the water through its lateral line system, and it consumes the amphipods it encounters. Its metabolism is so slow that a single feeding event may sustain it for days or weeks. Its life, by surface standards, is almost impossibly quiet, and yet this creature occupies the same ecological position at the bottom of the Mariana Trench that a great white shark or a killer whale occupies in surface waters. It is the end of the line, the final consumer, the organism at the top of a food chain that begins with the chemistry of the Earth itself. The entire ecosystem of the Hadal zone can be described in a sentence. Rock reacts with water, bacteria feed on the products, amphipods eat the bacteria and the detritus that falls from above. Snailfish eat the amphipods. That is the complete food web of the deepest place on Earth. 4 links. No more. A chain so short and so simple that it fits in the palm of your hand, and it has been functioning in essentially this form for millions of years. There is a kind of elegance in that simplicity. On the surface, ecosystems are tangled webs of interdependence, with thousands of species interacting in ways that ecologists are still struggling to untangle. In the trench, the web is reduced to a thread. Each organism depends on the one below it, and the one below that depends on the chemistry of the rock. If the rock stops producing hydrogen, the bacteria die. If the bacteria die, the amphipods starve. If the amphipods starve, the snailfish vanishes. The entire system is balanced on a single chemical reaction, a single geological process, a single principle. And it holds. It has held for longer than most surface ecosystems have existed. The Hadal Zone has been a stable environment for tens of millions of years, buffered from the catastrophes that periodically reset life on the surface. No asteroid impact, no ice age, no volcanic winter, no mass extinction has reached this deep. The food chain of the Mariana Trench is ancient, minimal, and remarkably durable. It asks almost nothing of the world above. It receives almost nothing from the world above, and it continues, uninterrupted, in the dark. There is one more layer to the story of the Mariana Trench, and it is the one that brings the narrative home. It is the layer in which we stop being observers and become participants, in which the trench, which has existed for 52 million years and hosted life that predates our species by orders of magnitude, meets the unmistakable signature of human civilization. On May 1st, 2019, a man named Victor Vescovo descended to the floor of the Challenger Deep. Vescovo is a retired United States Navy officer and a private equity investor, and he had spent several years and a considerable fortune building a submersible capable of reaching the deepest points in the world's oceans. The submersible was called the Limiting Factor, a titanium-hulled vehicle designed to withstand pressures exceeding those at the bottom of the Challenger Deep by a margin of 20%. It was, and remains, one of the most capable deep-sea submersibles ever built. Vescovo's descent was part of a larger project called the Five Deeps Expedition, an effort to reach the deepest point in each of the world's 5 oceans, the Atlantic, Pacific, Indian, Southern, and Arctic. The Mariana Trench, being the deepest of all, was the expedition's culmination. Vescovo and his team made 5 separate dives to the Challenger Deep during their time in the Mariana Trench. On the deepest dive, Vescovo's instruments recorded a depth of 10,927 meters, or 35,853 feet. This set a new record for the deepest manned sea dive in history, surpassing the previous record held by the filmmaker James Cameron, who had descended to approximately 10,908 meters in 2012. Vescovo spent 4 hours on the floor of the Challenger Deep. He moved slowly across the bottom, scanning the terrain with the submersible's cameras and lights. He observed several species of marine life, including amphipods, sea cucumbers, and what his team's scientists believed to be at least 4 previously undescribed species. He saw brightly coloured rocky outcrops that may have been the result of microbial activity on the seafloor. He documented the terrain, the water conditions, and the biology with the thoroughness of a scientist and the enthusiasm of an explorer. And among all of these things, at the very bottom of the deepest place on Earth, he found a plastic bag and candy wrappers. The discovery was not entirely surprising to the scientific community, though it was reported widely in the media because of the stark symbolism of the image. A plastic bag, the most mundane and disposable of human artifacts, sitting on the floor of a place that most of humanity will never see, at a depth that exceeds the cruising altitude of most commercial aircraft. It is, by any measure, a long way for a piece of trash to travel. Plastic in the ocean is not a new phenomenon. Scientists have been documenting the accumulation of plastic debris in the world's oceans since the 1970s. The numbers are staggering and, at this point, well known. Approximately 390 million tons of plastic are produced globally each year, and of that, an estimated 14 million tons find their way into the ocean. The The plastic arrives through rivers, through storm drains, through coastal dumping, through the breakdown of larger pieces of debris into smaller fragments. Once in the ocean, plastic does not biodegrade in the way that organic materials do. It does not decompose and return to the carbon cycle. Instead, it breaks down mechanically, fragmenting into smaller and smaller pieces under the influence of sunlight, waves and abrasion. These fragments, called microplastics, when they are smaller than 5 millimeters, can persist in the marine environment for centuries. What was significant about the plastic found by Vescovo, and about the research conducted in the same year by the marine ecologist Alan Jameson and his colleagues, was the depth at which it was found. The Challenger Deep is not a coastal environment. It is not a river mouth or a harbour or a beach where plastic debris might be expected to accumulate. It is the most remote point on the surface of the Earth, measured not by horizontal distance from human habitation, but by vertical distance from the surface. To reach the floor of the Challenger Deep, a piece of plastic must descend through nearly 11 kilometers of water. It must settle through the sunlit zone, through the twilight zone, through the midnight zone, through the abyssal zone, and finally through the Hadal zone, a journey that can take weeks or months, depending on the size, density, and buoyancy of the object. And yet it arrives. It arrives because the Mariana Trench, by virtue of its geometry, functions as a kind of funnel. The V-shaped profile of the trench channels material that falls from the overlying water column into the narrow valley at its base. Organic matter, sediment, the remains of organisms from the surface, all of these accumulate at the bottom of the trench in concentrations that are higher, in some cases significantly higher, than on the adjacent abyssal plain. This funneling effect is what sustains the surprisingly abundant communities of amphipods and other organisms at Hadal depths. It is also what concentrates human debris. In February of 2019, the same year as Vescovo's dive, Jameson and his colleagues published a study in the journal Royal Society Open Science that documented the extent of microplastic pollution in the deepest parts of the ocean. The team had collected 90 amphipods, small crustaceans of the suborder Lysianassoidea, from 6 deep ocean trenches around the Pacific Rim. The trenches included the Japan Trench, the Izubonin Trench, the Mariana Trench, the Kermadec Trench off New Zealand, the New Hebrides Trench near Vanuatu, and the Peru-Chile Trench off the west coast of South America. The amphipods were collected at depths ranging from 7,000 metres to 10,890 metres, the latter being the depth of the Challenger Deep itself. The researchers dissected each amphipod and examined its hindgut, the final section of the digestive tract, for the presence of microplastic particles. What they found was comprehensive and unambiguous. Of the 90 amphipods examined, 65 contained at least one microplastic particle, that is 72%. The number of particles per individual ranged from 1 to 8. The particles were identified using Fourier transform infrared spectroscopy, a technique that can determine the chemical composition of a material, and they were found to include nylon, polyethylene, polyamide, polyvinyl alcohol, and polyvinyl chloride, as well as semi-synthetic fibres like rayon and natural fibres like rami. The particles were tiny, measured in micrometers, but relative to the size of the amphipods that ingested them, they were substantial. The contamination was not evenly distributed across trenches. Some trenches showed higher rates of ingestion than others, but the Mariana Trench stood out. At Challenger Deep, the deepest site sampled, 100% of the amphipods examined contained microplastic particles, every single one. The mean number of particles per individual was also highest in the Mariana Trench, at 3.3 particles, compared to a low of 0.9 in the New Hebrides Trench. The researchers noted that the variation between trenches was likely related to differences in the overlying surface productivity and proximity to sources of plastic pollution. The Mariana Trench lies in the western Pacific, a region with heavy shipping traffic and downstream of some of the most densely populated coastlines on Earth. The New Hebrides Trench, by contrast, lies in a more remote area of the South Pacific with less direct exposure to plastic sources, but the overarching finding was clear. Microplastic pollution had penetrated to the deepest point on the planet and was being actively ingested by the organisms that live there. Jameson, in an interview with the Agence France Presse, put the finding in characteristically direct terms. The deep sea, he said, is the ultimate sink. Whatever enters the ocean will eventually settle, and once it reaches the Hadal zone, there is no mechanism to bring it back. The plastic that enters the trench stays in the trench. It does not wash ashore. It does not float back to the surface. It settles into the sediment, is ingested by amphipods and other organisms, and enters the deepest food web on the planet. The amphipods of the Mariana Trench evolved in a food-scarce environment. They are scavengers, adapted to eat whatever organic matter sinks to the bottom. Their feeding strategy is opportunistic and unselective. When a whale carcass or a mass of marine snow reaches the trench floor, the amphipods converge on it in swarms of hundreds, feeding voraciously until the food source is exhausted, then returning to a state of quiescence until the next meal arrives. This strategy, refined over millions of years of evolution, is extraordinarily effective for exploiting a sparse and unpredictable food supply. But it also means that the amphipods cannot distinguish between a particle of organic matter and a particle of plastic. Both are ingested with equal readiness. Microplastics are not merely inert. They can absorb toxic chemicals from the surrounding water, including persistent organic pollutants such as polychlorinated biphenyls, commonly known as PCBs. A separate study by Jameson and his colleagues found extraordinarily high concentrations of PCBs and other persistent organic pollutants in the tissue of amphipods from the Mariana Trench and other deep ocean trenches. The concentrations were, in some cases, comparable to those found in organisms living in heavily polluted estuaries on the surface. The deep ocean, which we tend to imagine as pristine, is accumulating the chemical residue of decades of industrial activity. Once microplastics enter the Hadal food web, they have no exit. Amphipods are consumed by other organisms. Those organisms are consumed in turn. The plastic fragments and the chemicals they carry move through the food chain, concentrating at each trophic level. The trench, which funnels material downward with remarkable efficiency, has no corresponding mechanism for exporting it. What goes down stays down. There is a deeper question embedded in the story of plastic at the bottom of the Mariana Trench, and it is not a scientific question. It is a philosophical one. We are still learning what exists in the deepest parts of the ocean. We have barely begun to catalogue the organisms that live there, to understand their biology, to map the ecosystems they form. Every expedition to the Hadal zone discovers new species. Every study reveals new adaptations, new chemistries, new relationships between organisms. The Mariana Trench is, by any measure, one of the least explored and least understood environments on Earth. And yet our presence arrived there before our understanding did. The plastic that Vescovo found at the bottom of the Challenger Deep was not placed there deliberately. Nobody intended to pollute the deepest point on the planet. It arrived through the ordinary unremarkable operation of global consumer culture. Someone somewhere used a plastic bag. It entered the ocean, perhaps through a river or a storm drain or the wind. It sank slowly, over weeks or months settling through the water column until it came to rest at a depth of nearly 11 kilometres. And there it sits, alongside the amphipods and the xenophyophores and the chemosynthetic bacteria, a durable artifact of a civilization that has existed for a few thousand years in an ecosystem that has been operating for tens of millions. The plastic will not disappear. Not in our lifetime, not in our children's lifetime. The conditions at the bottom of the Mariana Trench, cold, dark, devoid of the ultraviolet light that drives photodegradation at the surface, are nearly ideal for the preservation of plastic. It will persist for centuries at minimum. It will be ingested, fragmented and redistributed through the sediment and the food web but it will not decompose. It will remain. It is a small thing, a plastic bag at the bottom of the ocean. It is easy to know about it and move on, to file it under the general heading of pollution and return to the more compelling mysteries of deep sea biology and geology. But there is something about the image that resists dismissal. Perhaps it is the contrast between the antiquity of the place and the triviality of the object. A place that has existed for 52 million years, that hosts life older than the surface world, that resists our instruments and our understanding, and on its floor lies a piece of disposable packaging from a convenience store. The trench does not care about the plastic. It has no opinion about it. The amphipods eat it because they eat everything. The microbes will, in geological time, find ways to break it down. Perhaps. The sediment will bury it, eventually, and it will become part of the geological record. A thin, faintly anomalous layer in the stratigraphy of the trench, a signature of the brief period during which a particular species of primate dominated the surface of the earth and scattered its debris into every corner of the planet. Perhaps the most important question is not what lies on the trench floor, but whether this place needs us at all. The Mariana Trench has operated for 52 million years without human intervention. Life here evolved in darkness, in silence, under pressures that would crush our bodies and our instruments alike. It found ways to survive that do not depend on sunlight, on the atmosphere, or on any process connected to the surface world. It predates our species by millions of years. It may predate all surface ecosystems by billions. We are still learning what exists here, and yet traces of our presence arrived long before we did. There is a sound the ocean makes that most people have never heard and never will. It is not the crash of waves against a shore. It is not the song of a whale or the click of a shrimp colony or the groan of ice breaking apart at the poles. It is the sound of the ocean itself, the ambient background noise of water molecules in thermal motion, of distant earthquakes propagating through the crust, of currents sliding across the contours of the seafloor thousands of kilometers away. At the surface, this sound is drowned out by the noise of life and weather, the slap of wind-driven waves, the hiss of rain, the biological chorus of a billion organisms going about the business of survival. But as you descend, the noise of the surface world fades. By 1,000 meters, the biological sounds have thinned to isolated clicks and pulses. The conversations of deep-diving whales and the bioluminescent flashes of creatures that communicate in light rather than sound. By 3,000 meters, even these become sparse. By 6,000 meters, the ocean is quieter than almost any environment on the surface of the earth, quieter than a library, quieter than a recording studio, quieter than the inside of a cave at midnight. And at the floor of the Challenger Deep, the ambient noise approaches something close to the theoretical minimum. The background sound recorded by Deep Sound 2 before the implosion of its companion instrument was dominated by the very lowest frequencies, the deep, slow oscillations of geological processes and distant ocean events. It was a hum so low and so faint that it exists at the edge of what an instrument can detect and well beyond the range of human hearing. If you were to listen to it amplified and time-stretched into the range of human perception, it would sound like the breathing of the planet, a slow, steady, featureless exhalation, without rhythm, without variation, without end. This is the acoustic landscape of the deepest place on earth. Not silence exactly, because true silence does not exist in any physical medium. Water transmits vibration. The rock beneath the trench transmits vibration. Even the molecules of the water itself, jostled by thermal energy, produce a faint random noise. But it is as close to silence as the natural world allows. And for a human brain, which evolved in an environment saturated with acoustic information, that near silence is profoundly disorienting. Sound, we forget, is not just a source of information, it is a source of emotional regulation. The hum of traffic outside a window, the rustle of wind through leaves, the distant murmur of conversation in another room. These sounds are not processed consciously most of the time, but they anchor us in a world of other beings, other events, other lives proceeding alongside our own. Remove them, and the brain begins to feel not just alone but unmoored. The absence of sound is not experienced as peace. It is experienced as deprivation, and the brain responds to deprivation with anxiety, with hypervigilance, and ultimately, if the deprivation continues, with the generation of its own internal sound, the auditory hallucinations that submarine crews and polar explorers and long-distance cave divers have reported for as long as humans have ventured into environments that their evolution did not prepare them for. The Mariana Trench does not try to frighten us. It does not contain monsters, at least none that we have found. The organisms that live at Hadal depths are small, slow, quiet, and largely uninterested in anything the size of a human being. The amphipods eat dead things. The xenophyophores sit on the seafloor and filter. The bacteria metabolize hydrogen in the rock. None of them pose a threat to a human body enclosed in a titanium submersible. The danger of the trench is not predation. It is not even pressure, provided the submersible is designed to withstand it. The danger is the environment's absolute indifference to the needs of the human mind. It provides nothing that the brain requires to remain oriented, stable, and calm. And in the absence of those provisions, the mind begins to falter, not through weakness, but through the perfectly normal operation of a system that was never designed for this. There is a broader lesson here, one that extends beyond the trench itself and into the way we relate to the natural world more generally. We tend to frame the deep ocean, and extreme environments in general, as challenges to be conquered. We celebrate the explorers who descend to the Challenger deep. We admire the engineers who build the submersibles that carry them. We mark the records: deepest dive, longest time at depth, first solo descent. These are genuine achievements, and the courage and ingenuity required to accomplish them are real. But the trench has not been conquered by any of these expeditions. It has been visited, briefly, by small numbers of people in machines designed specifically to keep the environment at bay. No one has ever swum at the bottom of the Challenger deep. No one has ever breathed the water there or touched the sediment with bare hands. The submersibles that descend to the trench are, in the most literal sense, artificial environments, bubbles of surface conditions maintained at enormous expense against the crushing reality of the surrounding deep. The moment the hull fails, the moment the glass sphere cracks, the occupant ceases to exist in any form recognizable as human life. The environment outside the submersible is not survivable, not for a second, not for a fraction of a second. This is what distinguishes the deep ocean from almost every other extreme environment on Earth. Humans have climbed Everest without supplemental oxygen. Humans have crossed Antarctica on foot. Humans have survived for weeks in the open ocean on rafts. These are extreme achievements, pushing the limits of human physiology and endurance, but they are achievements within the envelope of conditions that a human body can, with effort and preparation, tolerate. The deep ocean is outside that envelope entirely. There is no amount of training, no level of physical fitness, no mental discipline that would allow a human being to survive unprotected at the bottom of the Mariana Trench. The gap between what we can endure and what the trench demands is not a matter of degree. It is a difference in kind. And this, perhaps, is why the trench fascinates us. It is one of the few places on our own planet that is genuinely alien. Not alien in the metaphorical sense of being unfamiliar or exotic. Alien in the literal sense of being incompatible with human life. A place where the physical conditions exceed the design specifications of the human body so completely that no adaptation, no technology short of a sealed vehicle can bridge the gap. The surface of Mars is hostile, but the hazards there, low pressure, cold, lack of oxygen, are hazards that a well-designed spacesuit can address. The bottom of the Mariana Trench requires not just protection, but total isolation from the environment. The organism inside the submersible must be kept in a separate physical reality from the water outside. We are visitors here, brief, tentative, and utterly dependent on our machines. The organisms that live at these depths are not visiting, they are home. They have been home for millions of years. The amphipod that eats at the bottom of the Challenger Deep is as much a native of its environment as a sparrow is of a garden hedge. It does not struggle against the pressure. It does not suffer from the darkness. It does not miss the sun. It has never known the sun. It exists simply and completely in a world that would destroy us. There is something both humbling and liberating in that recognition. Humbling because it reveals the narrowness of the conditions under which human life is possible. We inhabit a thin shell on the surface of a planet, breathing a specific mixture of gases, warmed by a specific range of temperatures supported by a specific range of pressures. Step outside that shell, whether into space or into the deep ocean, and we cease to be viable. We are, in the most fundamental sense, surface creatures, adapted to a thin, bright, temperate, oxygenated strip of the Earth's surface and nowhere else. But liberating too, because the trench shows us that life itself is not so constrained. Life has found ways to exist where we cannot. It has adapted to pressures that would crush our bodies, to darkness that would dissolve our minds, to temperatures that would slow our metabolism to a stop. It has found energy sources that do not depend on the sun, on the atmosphere, or on any process connected to the surface world. Life, as the Mariana Trench demonstrates, is far more resourceful, far more persistent, and far more indifferent to our definitions than we tend to give it credit for. The trench has been here for 52 million years. It will be here long after we are gone, and the life it contains, the snail, fish, and the amphipods and the bacteria metabolizing hydrogen in the dark, will continue, patient and unconcerned, in a world that was complete before we knew it existed. What the trench leaves us with, after all the science and the measurements and the images of plastic on the ocean floor, is not a conclusion. It is a collection of open questions, each one deeper than the last. And the answers, if they exist, lie in places we have barely begun to explore. We began at the surface looking down. We followed the light as it faded, watched the colours disappear one by one. Red, orange, yellow, green, and then blue, and then nothing. We descended into a world governed by pressure, where the molecules that make up living things are forced into configurations that our textbooks do not predict. We met the organisms that survive there. The translucent snailfish, with its 5 copies of a single essential gene. The amphipods that supplement their cellular chemistry with compounds borrowed from Alzheimer's research. The xenophyophores that have simplified life to its barest expression, a single cell the size of a fist, motionless on the floor of the world. We watched evolution run in reverse, shedding bone and pigment and sight and speed until only the essentials remained. We discovered ecosystems that owe nothing to the sun, powered instead by the slow reaction of water and rock, a form of energy production that may be older than photosynthesis itself, and that may exist not only on this planet, but on the icy moons of distant worlds. We confronted the unsettling truth about what happens to the human mind when every sensory anchor is removed, when the brain, deprived of light and sound and temperature variation and spatial reference, begins to generate its own reality in the void. We learn that we cannot agree on the depth of the deepest place on Earth, that after 150 years of measurement, the number still shifts with each new survey, each new instrument, each new set of assumptions about the speed of sound in water. We discovered that the trench is not a fixed feature but a living process, the visible surface of a tectonic engine that has been running for 52 million years and shows no sign of stopping. And we found at the very bottom the unmistakable evidence of human presence, a plastic bag, candy wrappers, microplastic particles in the digestive tracts of every amphipod sampled from the deepest site on Earth. Each of these discoveries changes something about how we understand the world. The pressure paradox tells us that life is not limited by the conditions we consider normal. It is limited only by chemistry. And chemistry, it turns out, has more flexibility than we assumed. Evolution in the trench tells us that complexity is not the inevitable direction of biological change. Under the right conditions, simplification is the more successful strategy, and the organisms that thrive are the ones that have learned to do less with less. The chemosynthetic ecosystems tell us that the sun, which we have treated as the indispensable foundation of all life, is in fact optional. Life does not require a star. It requires only energy, and energy can be found in rock and water and the slow heat of a planet's interior. The psychological effects of the deep tell us something about the construction of human consciousness that we do not normally confront. Our sense of self, our sense of orientation, our sense of time, all of these are not fixed properties of the mind. They are constructs, built in real time from the continuous flow of sensory data. Remove the data, and the constructs dissolve. The self is not a thing, it is a process. And like any process, it depends on inputs that the environment provides. The measurement problem tells us about the limits of human precision. We have sent robots to Mars and telescopes to the edge of the observable universe, and we cannot determine, to the nearest 50 meters, the depth of a hole in the floor of our own ocean. This is not a failure of technology or ambition. It is a reminder that some things are fundamentally difficult to know, that the physical properties of the medium, the speed of sound in variable density water, the irregular geometry of a narrow submarine valley, impose constraints that no amount of cleverness can entirely overcome. The tectonic story tells us about timescale. The Mariana Trench operates on a clock that measures in millions of years. A human lifespan is a rounding error in the history of the trench. A human civilization, from its earliest beginnings to its most recent moments, fits within a single geological blink. The trench has been moving at 60 to 70 millimeters per year for 52 million years, and it will continue for tens of millions of years more. Whatever happens to us, to our cities and our technologies and our debates about climate and politics and meaning, the trench will go on. The plates will keep moving, the deep will remain deep, and the plastic tells us about legacy. Not the legacy we choose, not the monuments and the books and the great works we hope will survive us, but the legacy we leave involuntarily, the residue of our daily lives, the bag that held someone's groceries, the wrapper that held someone's candy. These are the artifacts of our civilization that have reached the most remote point on the Earth's surface. They were not placed there with intention. They arrived through the ordinary operation of ocean currents and gravity, the same forces that have been delivering organic matter to the trench floor for millions of years. And they will remain there, preserved in cold and darkness, for centuries after the hands that used them have been forgotten. There is a temptation, at this point in the story, to make the trench into a metaphor, to say that it represents the unknown, or the unconscious, or the limits of human knowledge, or the fragility of the environment, or the hubris of technological civilization. And there is truth in some of those metaphors. The trench is, in a sense, all of those things, but it is also, and more fundamentally, just a trench, a physical feature of the Earth's surface, created by the motion of tectonic plates, inhabited by organisms that have adapted to its specific conditions, measured imperfectly by human instruments, and contaminated, in its upper sediments, by the waste products of a species that has existed for a fraction of its history. The trench does not mean anything. It does not symbolize anything. It does not carry a message or teach a lesson or point toward a moral. It is not a parable, it is a place. And the organisms that live there are not symbols of resilience or simplicity or the triumph of life over adversity. They are organisms. They eat, they reproduce, they die, they decompose, and the matter that composed them is recycled into the sediment and the food web and the slow chemistry of the deep. They do not know that they are remarkable. They do not know that we have studied them, named them, marvelled at their adaptations. They do not know that we exist. And perhaps that is the most important thing the trench has to teach us, even if it teaches nothing deliberately. That the natural world is not arranged for our benefit or our instruction. That it does not exist to inspire us or to humble us or to provide metaphors for our condition. It exists because the physics of the universe allow it to exist. It continues because the conditions that sustain it have not yet changed sufficiently to end it, and it will go on, regardless of whether we watch it or ignore it, understand it or misunderstand it, protect it or pollute it. The Mariana Trench was complete before we knew it existed. The organisms at its floor were thriving before our species emerged from the forest of East Africa. The chemosynthetic bacteria at Serena Deep were metabolizing hydrogen from serpentinized rock before the first human being looked up at the stars and wondered what they were. The tectonic plates that created the trench were moving before there was anyone to measure their speed. We arrived late. We arrived curious. We brought our instruments and our submersibles and, unintentionally, our trash. We measured and mapped and marvelled. And the trench received us with the same indifference it shows to everything, to the amphipods that feed on its floor, to the sediment that settles from above, to the rock that slides beneath it, to the water that presses on it from every direction, always, without pause, without variation, without end. If you have stayed awake through all of this, you have travelled in imagination, at least, to a place that fewer people have visited than the surface of the moon. You have descended through 11 kilometres of water and arrived at a floor that was ancient before the first grass grew on the continents above. You have met organisms that have no need of sunlight, no need of oxygen from the atmosphere, no need of anything that we would recognise as a hospitable environment. You have heard the only sound ever recorded at the deepest point on Earth, the implosion of a glass sphere, and you have learned that the echo of that implosion told us more about the depth of the trench than decades of deliberate measurements had achieved. And you have learned, if nothing else, that the deepest place on Earth is not empty, it is not lifeless, it is not dead. It is full, in its own way, of activity, of chemistry, of organisms pursuing survival under conditions that would seem impossible by any standard we apply on the surface. Life at the bottom of the Mariana Trench is quiet, slow, cold, dark, and utterly unlike anything we experience in our daily lives. But it is life, and it has been here, uninterrupted, for longer than we can easily comprehend. The ocean does not ask to be explored. It does not require our maps or our instruments or our names for the things that live inside it. It does not benefit from our attention or suffer from our neglect. It operates according to its own principles, on its own timescale, indifferent to the concerns of the brief, noisy species that has recently taken to examining it. The trench was here before us, it will be here after us. The organisms that call it home have no awareness of our existence, and no need of our understanding. They persist, because the conditions for their persistence are met. And when those conditions change, as they will, over geological timescales far beyond the scope of human history, the organisms will change too, or they will not, and the trench will continue either way, descending slowly deeper as the Pacific plate slides beneath the Philippine sea plate, millimetre by millimetre, year by year, epoch by epoch, until the tectonic forces that created it finally exhaust themselves and the Earth's interior cools, and the plates stop moving and the trench, at last, fills with sediment and disappears. But that is a story for a timescale we cannot experience. For now the trench endures, the water presses down, the bacteria feed on hydrogen in the dark, the amphipods scavenge in the sediment, the snailfish glides through water that has never seen the sun, and somewhere at the bottom, among the rocks and the mud and the quiet unceasing chemistry of the deep, there is a plastic bag, settled into the silt, waiting for no one, going nowhere, preserved in the cold and the pressure and the dark, a small, absurd monument to the species that sent it there. The deep does not judge us for it. The deep does not judge us for anything. The deep simply continues. And if you are still listening, and if the sound of these words has carried you somewhere close to sleep, then let the trench be the last image you hold. Not the plastic, not the pressure, not the darkness, but the continuity, the sheer unbroken persistence of life in a place that should, by every reasonable standard, be empty. The fact that chemistry found a way, that organisms found a way, that in the deepest, darkest, coldest, most hostile corner of this planet, something is alive, and has been alive, and will go on being alive, long after the surface world has changed beyond recognition. The deepest place on Earth was complete before we knew it existed. It will continue long after we're gone. It never needed us to understand it. And tonight, as you descend into sleep, the trench descends too, slowly, patiently, 1 millimeter at a time, into the dark, into the deep, into the long, quiet future that belongs only to the Earth. Rest now. The deep will still be here in the morning. It will still be here in 1,000 years. It has been waiting for 52 million, and it is in no hurry. Good night.